Assalamu alaikum everyone. I hope you all are doing great. So today the topic we are going to discuss is phenylalanine degradation and its related disorders. The disorder we are going to study is phenylketonuria. So first of all we will see what phenylalanine is. Here is a brief description of it. Phenylalanine. Phenylalanine, it is an aromatic and essential amino acid. So aromatic amino acids are those amino acids that contains aromatic ring. So this is structure of phenylalanine that shows that it contains an aromatic ring, an amine group, and a carboxylic acid group. It is an essential amino acid. So essential amino acid, these are those amino acids which cannot be synthesized from scratch in a body, or it cannot be synthesized fast enough to supply its demand in a body. So it must come from the diet. Therefore, phenylalanine is an aromatic and essential amino acid. So it is derived from artificial sweeteners such as aspartame and proteins. Phenylalanine, it is believed to be the precursors of many biologically important compounds such as tyrosine, dopamine, norepinephrine, epinephrine, and melanin pigment. This dopamine, it is a neurotransmitter, important chemical in the brain that helps to conduct nerve impulses or to take messages from one neuron to another. Norepinephrine and epinephrine. These both are hormones which are very important in your fight or flight response. And melanin pigment. This is a type of pigment that is present in the skin. It gives color to our skin, to hair, to our eyes. So phenylalanine, it is both glycogenic and ketogenic. So glycogenic amino acids are those amino acids that can be converted into glucose. Okay, the glycogenic amino acids are those amino acids that can be converted into glucose through gluconeogenesis. Whereas these ketogenic amino acids are those amino acids that are converted into ketone bodies. So phenylalanine can be converted into both glucose as well as ketone bodies. Therefore, it is said that it is both glycogenic and ketogenic. It is non-polar in nature. So non-polar amino acids that are those amino acids that have no polarity. So these amino acids have equal number of amine group and carboxylic group. As in the figure you can see that it has one amine group and one carboxylic group. So this amine group has positive charge, carboxylic group has negative charge. So the total charge is neutral. There is no charge on its side chain, no charge on its R chain. Okay, R group. Therefore, this is non-polar amino acid. And non-polar amino acids are hydrophobic in nature. And at last, it is encoded by UUC and UUU. So first of all, we will study the normal pathway, like how phenylalanine it is converted, it is degraded, okay, in our body. So as I have told you that it is an essential amino acid, so it must be taken from a diet. So the first step into the degradation of phenylalanine is that we take phenylalanine from a diet, okay. So phenylalanine, it is converted into tyrosine with the help of the enzyme phenylalanine hydroxylase, also written as PAH. Okay, so the function of phenylalanine hydroxylase is that it catalyzes the hydroxylation of this phenylalanine. As you can see that this is aromatic ring, this is amine group, this is carboxylic group. Whereas the tyrosine, there is the hydroxylic group. Okay, but here in the phenylalanine there is no hydroxylic group, which means that this phenylalanine hydroxylase, it catalyzes the hydroxylation of this aromatic side chain of phenylalanine and it generates what? Tyrosine. So the cofactor of this enzyme phenylalanine hydroxylase PAH is tetrahydrobiopterin, also written as BH4. Okay. So during this hydroxylation reaction in which the phenylalanine is converted into tyrosine, okay, this tetrahydrobiopterin, okay, this tetrahydrobiopterin BH4, it is oxidized to dihydrobiopterin, that is BH2, and then it again BH2 is then reduced to BH4, okay, with the help of this enzyme, that is dihydrobiopterin reductase enzyme. So this step that I have told you, that this is the first step of degradation of phenylalanine. This is the normal pathway in which phenylalanine undergoes various steps which involves various enzymes, cofactors, substrate, and it then finally converts phenylalanine into acetoacetyl-CoA. So this is the first step that I have told you, that phenylalanine, it is converted into tyrosine with the help of the enzyme phenylalanine hydroxylase, and the cofactor is tetrahydrobiopterin, that is BH4, okay, and then this BH4 is oxidized into BH2, and then this BH2 is then again reduced to BH4 with the help of, in the presence of this NADH. So this reaction requires NADH. Once this aromatic ring is hydroxylated, phenylalanine is converted to tyrosine. Then other enzyme, tyrosine aminotransferase, converts this tyrosine into para-hydroxyphenylpyruvate. Okay, it means that this aminotransferase enzyme, it transfers the amine group and it converts this alpha ketoglutarate to glutamate. Okay, here there is amino group, that is NH3 amine group, and here the amine group is removed and it converts that it is para-hydroxyphenylpyruvate. Then it is converted to homogentisate with the help of enzyme para-hydroxyphenylpyruvate dioxygenase. So di means two. So the function of the dioxygenase enzyme is that it incorporates both this oxygen atom into this substrate, okay, and it converts this para-hydroxyphenylpyruvate into homogentisate, okay, as the carbon dioxide molecule will be removed, it will be given off. Then this homogentisate, then again homogentisate 1,2-dioxygenase enzyme will came. It will then add another oxygen atom into the first and second position, and it will then cleave this aromatic ring and convert it into linear structure, that is malonyl acetoacetate, and then malonyl acetoacetate will be converted. Then this malonyl acetoacetate will be converted into fumaryl acetoacetate, which is its isomer with the help of enzyme malonyl acetoacetate isomerase. So the function of isomerase enzyme is that it catalyzes the reaction which involves structural rearrangement. As you can see that the hydrogen atom here, the both are in downward direction, whereas here the structurally rearranged, this malonyl acetoacetate is structurally rearranged and it is converted into fumaryl acetate. Okay, the whole structure is same, but here only there is a structural rearrangement in malonyl acetoacetate, which converts it into fumaryl acetoacetate, and then hydrolysis reaction take place where fumaryl acetoacetate, it will break down this fumaryl acetoacetate into two products, fumarate and acetoacetate. Okay, then this acetoacetate with the help of enzyme 3-ketoacyl-CoA transferase. So as name indicate that it will transfer the CoA group from succinyl CoA. It will transfer the acetyl group. It will transfer the CoA group from acetoacetate to the this fumaryl acetoacetate will be converted into two products. One is fumarate and other is acetoacetate. This acetoacetate will then be converted into acetoacetyl-CoA with the help of enzyme 3-ketoacyl-CoA transferase. It will actually transfer this CoA group from succinyl to the acetoacetate and it will make acetoacetyl-CoA. Okay, like here this S-CoA will be incorporated and this succinyl CoA will be converted into succinate. So what happens is in phenylketonuria, like as I've told you that in the first step of phenylalanine degradation, the phenylalanine is converted into tyrosine with the help of the enzyme phenylalanine hydroxylase. Okay, and this is its cofactor, that is tetrahydrobiopterin, which is converted into dihydrobiopterin, and this cofactor is required for the proper functioning of the enzyme, so that can this step like phenylalanine must be converted into tyrosine. So if there is deficiency in PAH gene, in this gene, phenylalanine hydroxylase gene, that controls this activity of the enzyme phenylalanine hydroxylase enzyme. If this enzyme is deficient, or if there is less amount of the enzyme is not there, obviously if there is no enzyme, then how phenylalanine can be converted into tyrosine? So phenylalanine will not be converted into tyrosine, it will not be broken down into tyrosine. Okay, the whole degradation pathway will not take place. Why? Because phenylalanine hydroxylase, the enzyme is not present. Okay, then in turn this phenylalanine will get accumulated in our body. It will get accumulated in our blood, in our tissues, okay, in our plasma, and as well as excreted in the urine. So this condition is known as phenylketonuria, in which there is an excess amount of phenylalanine in the body. Okay, so there are two conditions, like when phenylalanine hydroxylase enzyme is not present, and obviously phenylalanine cannot convert into tyrosine and it will get accumulated. And if this enzyme is present, the gene is working properly, there is an adequate amount of this enzyme, but its cofactor is not present. The cofactor is not working properly, it is malfunctioning. This is tetrahydrobiopterin is not working properly. If cofactor is not working properly, then enzyme will not work properly. Then again in that case, phenylalanine will not be converted into tyrosine, and then it will be accumulating in your body. And this will also lead to phenylketonuria. Like this, if the phenylalanine hydroxylase is not present, then it will lead to phenylketonuria. And again, if homogentisate 1,2 dioxygenase enzyme is not present, if it is deficient and if it is in small amount, then homogentisate will not be converted into acetate. Okay, then this whole step will not take place. Okay, then this condition will lead to alkaptonuria, in which there is an accumulation of homogentisate. In alkaptonuria, there is accumulation of homogentisate because this enzyme is not working properly, and this step will not take place. Homogentisate will not be converted into malonyl acetoacetate. Whereas in phenylketonuria, the phenylalanine hydroxylase is not working properly. Therefore, the phenylalanine will not be converted into tyrosine. So, phenylalanine will be accumulated in the body. But today's lecture we are going to cover only phenylketonuria. So we will be focusing on this step, this enzyme phenylalanine hydroxylase and its cofactor tetrahydrobiopterin. It will lead to phenylketonuria. So what happens that if the enzyme is deficient, PAH is not working properly, means that phenylalanine is not be converted, it is not converted into tyrosine. So what happens is that a small pathway initiated on site. Another pathway initiates in which this phenylalanine, with the help of enzyme aminotransferase, it will be converted into phenylpyruvate. Okay, it will be converted into phenylpyruvate, and then this phenylpyruvate will then undergo decarboxylation, and it may produce phenyl acetate, or it may undergo reduction, and this phenylpyruvate may produce phenyl lactate. Okay, so the level of phenylalanine will increase, then other small pathway will initiated, it will produce phenylpyruvate, the level of phenylpyruvate will increase, the level of phenyl acetate will increase, the level of phenyl lactate is increased. So these all are ketone bodies. Okay, these all are ketone bodies. So this minor pathway will get initiated. Therefore, this phenylalanine and the phenylpyruvate, it will accumulate in our blood, in our tissue, and then the rest will be excreted out in the urine. Hence, this condition is named as phenylketonuria. This phenylpyruvate is either decarboxylated to form phenyl acetate or phenyl lactate. The production of phenyl acetate will result in the characteristics odor, that is mousy odor, in the urine. Okay, so these ketone bodies, that is phenyl acetate, when it will be excreted in the urine, it will give urine its mousy odor, which is one of the characteristics or symptoms of the person suffering from phenylketonuria. So these ketone bodies, they can also interfere with the blood-brain barrier. It can also lead to mental retardation. So phenylketonuria, in classical phenylketonuria, there is deficiency of this enzyme, that is phenylalanine hydroxylase. So if the level of the pH is normal, but the enzyme will not work properly without the BH4. Okay, if the enzyme is not working properly, then phenylalanine will be accumulated. If its cofactor BH4 tetrahydrobiopterin is not working properly, then obviously again phenylalanine will be accumulated in your body. Hence phenylalanine gets accumulated. So phenylketonuria, it is an inborn metabolic disorder in which there is mutation in PAH gene, that is phenylalanine hydroxylase gene, which will result in the low level of phenylalanine hydroxylase enzyme, and then which causes the dietary phenylalanine to build up to toxic level. This is known as hyperphenylalanemia, in which there is a high level of phenylalanine in the body. Untreated PKU can lead to intellectual disabilities, seizures, and mental disorder. Babies or affected mothers face heart problems, small head, low birth weight, and musty odor of urine and sweat. During pregnancy, mother's body is able to metabolize phenylalanine. Okay, when the mother is pregnant, she can able to metabolize this, break down this phenylalanine. That's why the infant with PKU, they have normal birth genetics. So moving on towards genetics, it is an autosomal recessive disorder, which means that both the copies of gene must be defected for the disease to develop. If the both parents are carrier, then there will be 25% chances that child will be born with PKU, 50% chances that child will be a carrier, whereas 25% chances that child might not be the carrier nor be affected by PKU. It affects both male and female equally. So this is the autosomal recessive disorder in which both the parents, they are carrier, they have one normal gene and the other one is defective gene. Okay, so these are the chances of a child, whether it is unaffected carrier child, unaffected carrier child, and the 25% chances that child is affected, 50% chances that this is unaffected carrier child, and 25% chances that it is a healthy child. Types of PKU. So there are three types of PKU. One is the classical PKU, in which the enzyme PAH is deficient, less amount are absent. So patient with blood phenylalanine concentration greater than 1200 micromole per liter are considered to be suffering from classic PKU. If this order remains untreated, it causes intellectual disabilities, seizures, and other serious health problems. Other type is variant PKU or non-phenylketonuria hyperphenylalanemia. It is rare condition in which different kind of mutation in PAH gene resulting in overfunctioning of PAH. In classical phenylketonuria, PAH is not working properly. Okay, it is in less amount. It is downregulated. Whereas in variant phenylketonuria, there is an overfunctioning of PAH enzyme. It means that more and more and more phenylalanine will be degraded into its product. Okay, so it means that phenylalanine in this case, it will not get accumulated in the body. So such patients are at the smaller risk of brain damage and don't even require treatment with low phenylalanine diet. The last one is hyperphenylalanemia due to deficiency of cofactor BH4. As I've told you that if the cofactor is not working properly, but the enzyme is normal, then again the phenylalanine will not be converting to tyrosine. So BH4 is a cofactor of PAH. Even if pH level is normal, the enzyme will not function without BH4. So if BH4 is malfunctioning, phenylalanine will still get accumulated in the blood. In such cases, if the patient is misdiagnosed as a PKU patient, they will suffer neurologically as they have normal amount of plasma phenylalanine. Hypopigmentation. First of all, this. As I have told you that phenylalanine is converted into the tyrosine, the enzyme phenylalanine hydroxylase enzyme. So in the classic phenylketonuria, the phenylalanine hydroxylase enzyme is not present, which means that this enzyme is defective, which means that phenylalanine will not be converted into tyrosine, or it will be less phenylalanine is converted into tyrosine. So no or less tyrosine is required, which means that no or less dopamine will be produced. No or less dopaquinone is produced. Similarly, no or less melanin will be produced. So that's why the patient suffering from phenylketonuria, they have lighter skin because they have no the melanin pigment is in very small amount, or it is completely absent. Okay, that's why the patient suffering from phenylketonuria so also suffer from hypopigmentation. So tyrosine is a precursor for synthesis of melanin. If this enzyme is not working properly, then tyrosine level drops, and as a result melanin is produced in PKU patient, less melanin produced. This is a result of light skin of PKU patient. Mousy odor. So as I have told you that the level of these ketone bodies, that is phenyl lactate, phenyl acetate, phenyl pyruvate, increases. Okay, because that short pathway gets initiated when phenylalanine gets accumulated. So these substances gives urine a mousy odor. Therefore, the disease is called as phenylketonuria because one of the dominant metabolites contain ketone groups, that is phenylpyruvate and alpha keto acid. This is another short pathway in which it shows that from our diet we take phenylalanine. If the enzyme is not present, phenylalanine hydroxylase is not working properly. If its cofactor is not working properly, then obviously phenylalanine will not be converting to tyrosine. Then a short pathway will get initiated in which this will be converting to ketone bodies, phenylpyruvate, phenyl lactate, phenyl acetate. And then at last mental retardation. So when passing through blood-brain barrier, phenylalanine will build up in tissues and hinders the pathway for other large neutral amino acids such as tyrosine and tryptophan, which are essential for protein and neurotransmitter synthesis. This phenylalanine accumulation will result in restricted development of brain, causing mental retardation. Symptoms: newborns affected with PKU do not show symptoms. They develop signs after a few months if they are not treated. So these following are the symptoms of phenylketonuria: musty odor in breath, urine, sweat, neurological problems including seizures, eczema, skin rashes, fair skin, blue eyes because phenylalanine is not converted into melanin, abnormally small head, microcephaly, intellectual disabilities, and delayed development. Diagnosis. Diagnosis of PKU is done via newborn screening. It is recommended to take the blood for screening after the first 24 to 28 hours of life after feeding proteins to reduce possibility of negative false results. If results come positive, then quantitative measurement of phenylalanine plasma should be done immediately. The method of choices are Kurzweil test, bacterial inhibition assay, tandem mass spectrometry identifies all the forms of hyperphenylalanemia with excellent precision. And at last, treatment. The PKU treatment comprises of low phenylalanine diet. Obviously, if our body is not able to break down the phenylalanine, so we must not be we must not take it from our diet, and monitoring the blood level of phenylalanine to ensure that it remains within normal levels for the first 5 to 10 years of life. Food high in phenylalanine must be eliminated from the person diet like meat, chicken, fish, eggs, milk. As I have told you that phenylalanine it is derived from the artificial sweeteners, aspartame. So aspartame is usually used as substitute for sugar. Aspartame is made up of two natural amino acids, that is L-phenylalanine and L-aspartame. So therefore, patients suffering from PKU must avoid sweeteners, aspartame, as it contains phenylalanine. Okay. So thank you so much. That it's for today lecture. Thank you so much. Allah Hafiz.